Hello everyone, welcome back to my channel. Today I'm going to talk about ionotropic glutamate receptors, and further in the ionotropic glutamate receptors I will talk about non-NMDA receptors and NMDA receptors. So the non-NMDA receptors are EMPA and kainate receptors, and I will talk in detail about the NMDA receptors, their function and their important properties. So let's get started. So this slide is showing you the classification of glutamate receptors. Glutamate receptors can be classified into ionotropic glutamate receptors and metabotropic glutamate receptors. In another video prepared by me, I have talked about the metabotropic glutamate receptors. So in this video, we will be focusing on the ionotropic glutamate receptors. So ionotropic glutamate receptors can be given the name as NMDA receptors and non-NMDA receptors such as EMPA receptors and kainate receptors. So these are basically given the name according to the pharmacological stimulation of these receptors with these agonists. So these are their subunits shown here. They are composed of different subunits, and the stimulation of the ionotropic results in the fast excitatory postsynaptic response, or in other words, we can say that the fast excitatory synaptic transmission. So further looking at the structure of the non-NMDA receptors, non-NMDA receptors we have already discussed are EMPA and kainate, and their structure of this ion channel is shown here. It's a kind of like simple as compared to the NMDA ion channel, which is complex, modulated by so many other substances. So when this ion channel or the glutamate receptor behaves like a simple channel, where the opening of this channel causes binding of the glutamate, results in opening of this channel, and there is movement of sodium inside the cell, and there is excitatory postsynaptic potential. So similarly, the NMDA receptor is also permeable to sodium, but it is more permeable to calcium as compared to the non-NMDA or EMPA receptors. So the metabotropic glutamate receptors, their functional anatomy is shown here. So whenever they are stimulated, they result in the stimulation of G protein. So glutamate action on the ionotropic receptors is always excitatory. So this is an important point to be taken into the account. So further, talking about the non-NMDA receptors, the excitatory synapses have both non-NMDA and NMDA receptors. At the normal resting potential, the non-NMDA receptors generate the large early component of the EPSP in the neurons in response to the stimulation. So these receptors get ion channels. It means that the sodium moves inside the cell. In the cell when there is stimulation of these receptors as a result of binding of the glutamate at its site. So they have relatively low conductance. Low conductance means compared to the NMDA receptors, these non-NMDA receptors have low conductance, that is less than 20 picosiemens. That is, and they are permeable to both sodium and potassium, but usually less permeable to the calcium. And whereas NMDA receptors are more permeable to the calcium ion than the sodium ions. So as a result, whenever there is stimulation of the NMDA receptors, large amount of calcium moves inside. So now let's talk about the NMDA receptors. Their structure is shown here. It is quite complex, modulated by so many things. But most important point is that under normal conditions, the magnesium is present. There is a magnesium binding site here, which results in the blockade of receptors under normal condition. NMDA receptor is basically having three very important properties. What are those properties? First one, the receptor controls a cation channel of high conductance that is permeable to calcium as well as the sodium and potassium. It means whenever there is stimulation of this receptor, there is large amount of calcium which will move inside the cell, and it will cause the stimulation in the postsynaptic receptor. The opening of the channel requires extracellular glycine as a cofactor. So glycine is shown here. It will bind at the site present on the NMDA receptors. Under normal condition, concentration of glycine in the extracellular fluid is sufficient to allow the NMDA receptor channel to function efficiently. Third, the channel is unique in a sense that its opening depends upon the membrane voltage as well as the chemical transmitter. It means that for the opening of NMDA receptors, it is important that the membrane is depolarized and glutamate is present and bound on the receptor. So this will cause the opening of NMDA receptors. So NMDA receptor opening will cause large amount of calcium to move inside the cell, and that will actually make a difference whenever there is NMDA stimulation and non-NMDA stimulation. So in this slide, the NMDA current is shown at three different membrane voltages at -80, -40, +20. So APV is the blocker of NMDA receptors. At -80, in the presence or absence of NMDA blocker, there is no difference in the current. It means that the current is at this point, the NMDA component is minimal. At -40, so just keep in mind that at the excitatory synapse, whenever glutamate is released from the presynaptic terminal, both non-NMDA and NMDA receptors are stimulated. So this is basically the cumulative response of the NMDA and non-NMDA shown, and it is actually distinguished by the presence of APV. So at -40, so this component, this is the graph without APV, and this is the current graph with the presence of APV, which blocks NMDA receptors. It means that this much component of the current is contributed by the NMDA. At +20, so you can see that the large amount of NMDA component is contributed, because at this time point magnesium is removed and large amount of current is actually generated by the NMDA receptors. So one more thing over here, you can see this line which is actually representing 25 millisecond. It means that the NMDA component is more prominent in the late component of EPSP after 25 milliseconds. So you can see that the NMDA component is more prominent and mostly present in this part of the EPSP. That is why we call it that NMDA contributes to the late component of the EPSP. So so one more thing is that, so from -40 to +20, you can see that there is a change in the direction of the current. This is because of the reversal potential. So reversal potential for both of these receptors is 0 millivolts. So other important properties of the NMDA receptors, they are that the NMDA receptors activated channels and extrinsic blocking by the magnesium is done. And magnesium is there. So this causes the blocking of the channel, and current flow at the normal basic synaptic transmission is minimal through the NMDA receptors. But when the membrane is depolarized by the action of glutamate, magnesium is expelled from the channel by electrostatic repulsion, allowing sodium and calcium to enter inside the cell. Maximal current flow through the NMDA channel takes place only when two conditions are met. Glutamate is present and cell is depolarized. NMDA receptor has further interesting properties, that it is inhibited by the hallucinogen drug phencyclidine and by the MK801, both of which bind at the site when the open channel pore is site within the open channel pore that is distinct from the magnesium binding site. You can see that the phencyclidine and like the other modulators, they bind at the different sites. Further, NMDA channel opens and closes relatively slowly in response to the glutamate. So the kinetics of the NMDA receptors are relatively slow. So, and it contributes to the late phase of EPSP that we have discussed. Because NMDA receptors require a significant level of presynaptic activity before they can function maximally. So long-term synaptic modification mediated by the NMDA receptors is often referred to as activity-dependent synaptic modification. Remember that the Non-NMDA receptors does not allow too much of the calcium to move in, but whenever there is stimulation, regulated or long-term stimulation of the synapse, so this will cause opening of the NMDA receptors and large amount of calcium will move in, and it will act as a secondary messenger, and it will cause synaptic modification or synaptic plasticity, and this is known as activity dependent synaptic plasticity. Okay, so that was it for the time. Thank you so much. Have a nice time.